Hi, good evening, everyone. I want to welcome you to Hudson Alpha, and thank you for being here for our annual Memory and Mobility Update. My name is Russell Smith. I'm the Chief Operating Officer at Hudson Alpha, and I joined about 18 months ago. I joined Hudson Alpha because I wanted to be part of an organization that's committed to make a difference in lives. Hudson Alpha is this unique and special place where discovery meets purpose. And I see every day how our teams turn genetic discoveries into basically impact, real life that matters. Faster ability to more quickly diagnose disease, crops that are more resilient and stronger, biotech innovation that fuels economy and impacts lives. And so again, I'm honored to be a part of Hudson Alpha and a group that's committed to make a difference. I know you, like me, are here tonight for the Memory and Mobility Update. And for my wife, Michelle, and I, this hits close to home. We certainly have seen the impact of neurodegenerative disease in our family. I won't poll hands, but I bet if I did, a lot of you are likewise impacted, either through family members or close friends or acquaintances. But likely many of us have seen the impact of neurodegenerative disease in our families and loved ones. We've got a team of scientists here tonight that have committed their careers. They wake up each day to advance research in Alzheimer's disease and related dementias, ALS and Parkinson's. Tonight you'll hear about new exploration in areas of what changes in DNA, how that might impact brain inflammation. You'll get updates on our Hope AD. I think all of you have probably heard about Hope AD and are likely looking forward to hearing updates on that tonight. You'll have a chance to ask questions and to be able to have Q&A with our scientists and with our team. But before we get into any of that, I just want to take a moment to thank our sponsors. Herb and Terry Lewis, longtime friends and gracious donors at Hudson Alpha. And I want to personally thank you. You were some of the first that I met at Hudson Alpha. You have been a great friend and supporter to me, and thank you for that. And I also want to recognize Rob Warren, a partner and wealth advisor at Twickenham Advisors. And so thankful for the partnership with Twickenham Advisors. They have been a longtime sponsor of our Memory and Mobility event. And I want to invite Rob to the stage to share a few words before we kick off.
Thank you all so much for coming and supporting such a wonderful event. As Russell mentioned, unfortunately, the topics that we'll learn about tonight is something that affects almost everybody in this room. I'm sure we can all raise a hand and we know a loved one or a friend or a family member that's been affected by neurodegenerative disease, and that's exactly how we got involved. And we've been a longtime supporter of Hudson Alpha and specifically this event because of the impact that we've seen on our clients and our friends and family. So on behalf of the rest of our team that's still back in the office, we thank Hudson Alpha, we thank you guys for attending tonight and look forward to hearing and learning about the incredible updates that have occurred over the last year. So thank you very much.
Thank you, Rob and Twickenham Advisors and Herb and Terry. And now I'm going to invite Sarah Sharman to the stage. Sarah is our Director of Communications, and look forward to the program and the team tonight. So thank you.
Good evening. It is so great to see a room full of people excited to hear about the progress being made in the field of neurodegenerative disease research. As Russell mentioned, I have the pleasure of serving as the Director of Communications here at Hudson Alpha. This is my 6th M&M update since I started at Hudson Alpha and my 4th year moderating the event. Every year, I look forward to hearing what our researchers have done to make a tangible impact on these diseases. My graduate school roommate studied Alzheimer's disease almost a decade ago, and the progress that has happened since then is really, truly inspirational. We now have a few disease-modifying monoclonal antibodies that can clear some of the damaging plaques and tangles in the brain and slow disease progression. We have reliable, accurate blood-based biomarkers that can detect Alzheimer's disease before symptoms have appeared. And we've made immense progress in understanding the underlying genetics and risk factors of these diseases. HudsonAlpha is at the forefront of this field, but we still have a long way to go. Today, current research is turning to the question of how do we detect and prevent these diseases before the devastating symptoms appear. Our teams are right in the thick of it, looking for new genetic factors, biomarkers, and environmental and lifestyle influences that can truly move the needle, not only on Alzheimer's disease, but other neurodegenerative diseases like Parkinson's, ALS, and Huntington's disease. My main passion as a science communicator is to help people understand the impact of our work and how it affects their lives. Part of that means listening to you all and hearing your interests and concerns. Last year at this event, we took audience questions and asked our scientists to answer them live at the end of the evening. It was such a success that we decided to bring it back this year. To submit a question for the live Q&A portion, write it in legible handwriting on that note card you were handed at the door. We can't ask a question we can't read. And pass it to your left when I call for questions, and our lovely helpers will pick up your questions. And we will ask them of our scientists at the end. There will be 3 opportunities to submit questions, so don't feel like you have to rush and write it down when I ask the first time. Now, without further ado, let's get the people on stage you really want to hear from. Our first speaker tonight is HudsonAlpha's Chief Science Officer, Dr. Rick Myers, who will give you all an update on some of our biomarker work.
Thank you, Sarah. Nice to see all of you. I didn't see how many when I was sitting up front. Great to see you here. We've been doing this for a long time. And I think some of you may have heard me say this recently. This last 2, 3, 4, 5 years, I think we have more hope than we've ever had because of the advances that are being made in the field. I personally, like probably maybe all of you in this room, affected by it. My mother had Alzheimer's disease and my father had Parkinson's disease. I don't know if I should say this, but I started working on them before they were diagnosed with the disease, working on these 2 diseases and Huntington's disease. So I've decided I'm not going to work on any new diseases for now, for the sake of my loved ones. But so what I'm going to just tell you about one of our projects that we're doing. We have a bunch of things. You're going to hear some really exciting stuff from Ashlyn and Nick in a few minutes. Let's see if I got it here. So some of you have heard about this because we've been talking about it for a couple of years. We started working on it maybe 3 years ago. And then we but it's really come to essentially final on one of the diseases and just starting on one of the other ones. I do want to give credit to Ben Henderson. He was a senior scientist in my lab who moved to France last week with his lovely wife. He's been with us a long time. Ben's just a wonderful scientist, and we're continuing some of this work by interacting with him, but that's a picture of him there. So what we want to do is to develop a blood test for diseases, and what that means is kind of like we do for cholesterol. And we call these biomarkers. When you find something somewhere, could be in the blood, it could be elsewhere, that is an indication that you might have something. PSA for prostate cancer is one of those. It's not a great biomarker, but it is used. We started working on these years ago, initially with colon cancer, actually, but I've worked on neuro diseases for a long, long time, really wanted to see if we could apply them to this. So why do you want a blood test? I hope you can see this. I should have made the font bigger. You want to detect things early if you can. It doesn't matter what the disease is. The earlier you detect it, the earlier you have a chance to do something about it. And it's really true in cancer. It's true in these disorders as well, before symptoms even start. Actually, one that people might not think about, you're trying to develop companies and academia trying to develop new treatments for something, you need to know whether the treatment in a clinical trial is working or not. And if the measures of how it's working are subjective, it's much, much harder to know how well it's working. So this may be the most valuable part of it, is to be able to use it in that way, use the blood test in that way. Wouldn't it be good if we could say this is Parkinson's or Alzheimer's or ALS or something, because sometimes the symptoms look very similar, or maybe something that we haven't even seen before. So to be able to differentiate between diseases, and I'm happy to tell you that we can on one of these as well now. And actually, and Nick Cochran may speak a little bit about this, but we can detect Alzheimer's disease now long before the symptoms if you do a very expensive imaging test. There are now biomarkers, you're going to hear about it as well, at least for Alzheimer's. We're trying to do that for ALS and for Parkinson's, and ultimately others as well. So the basis of a blood test is you got all these different. Am I the only one hearing that? Where? Oh, okay. Well, let me just try this. If you look at our brains, are the most complicated organ in our body by far. It has huge numbers of cells, trillions of cells, and a lot of different cell types. And that's just a cartoon of some of the cell types, and neurons and something called microglia and other types of cells. What happens is cells die anyway. The stuff in the brain doesn't usually die because it doesn't get replaced easily. But when you have a neurodegenerative, that's the word, means these cells are dying. When they die, they spill their insides out into the bloodstream. And especially in neurodegenerative diseases, this happens in other diseases as well. And what happens is the contents of the cells end up in the bloodstream, and that's where proteins, DNA, RNA, other stuff comes through your bloodstream. So taking a blood test, while it's not great to be stuck by a needle, that's a whole lot better than a lot of other invasive things that you might want to do. So that's the principle. You can look for DNA, RNA, protein. We're actually doing all three of those for Parkinson's disease, as you'll see. But for the ALS study that we did, we actually started looking for something they're called small RNAs. RNA is DNA's sister. RNA is actually made as a copy off of the DNA in little snippets, and we take particularly small snippets. That's actually about the length of it. It's about 22, 23 of these units of RNA and DNA. And so that's what we measure. And it had not been used as biomarkers when we started this study. It's starting to be now. So how you do this is you want to do a study. We want to say, let's just take ALS for the first one. Can we detect something in the bloodstream that someone with ALS has that people without ALS don't have? That it's as simple as that. It's really, and so you recruit participants, people with the disease and people without the disease. You like to get as many as you possibly can. I'm going to talk a little bit about the numbers in a bit. And then you take blood from them. They come into the clinic, or as you'll see, another clinic that we worked closely with throughout this time. And then so you draw that small amount of blood. It's a technical point, but you centrifuge it down and then take the clear stuff mostly, which is the plasma. And then what you do is you purify the RNAs from that plasma, and you run them through these sequencing DNA and RNA sequencing machines that we've been talking about we've been talking about for 35 years, but talking about since the day we got here, because that's what we do a lot of. It's not the only thing we do, but we do a lot of sequencing. And that actually then and you're sequencing this whole population of all this stuff in the bloodstream. You don't know what's there. You then look, and you can use computers to say, what are those, don't look at that yet, sorry. You say, what are all the different populations and the numbers of each one of those species of small RNAs are there? I'm moving. am I going? Oh, I'm going forward. I meant to be going backwards. Sorry. There we go. And it turns out there's a lot of analysis that you need to do to then figure out, to be able to essentially count what's there and then compare cases and controls. Controls means you don't have the disease. Cases means they have the disease. And I'm not getting, there's a bunch of statistical stuff that you do to then figure out am I really detecting? How good is the detection that I'm looking at? So we did this first one for ALS, and some of you know Sherry Kay. She's just an amazing, dynamic, wonderful person at Crestwood who worked with us from day one on this, collecting the, ALS is a little bit, it's rarer than many, certainly rarer than Alzheimer's, but people come from all over the Southeast and really all over the country to the center that they have at Crestwood because it's called a Center of Excellence. So she's been wonderful to work with. We really have loved working with her. And the initial study, we got a bunch of people who did not have the disease and blood from from 32 who did. That's not nearly enough to do the whole study. We then followed that up with more than twice that number in the follow-up. And what we did was, here it is. I'm sorry I'm going so fast. This thing has forward and backwards in different places. My fault, folks. No, usually I'm used to looking at it a different way. All right. So what happens is that we found one specific small RNA that was actually in the literature but had not been demonstrated as this. Ben found one that was elevated in people with ALS and not in people without ALS. And the statistics were very strong, and it replicated in that replication study, which was really important. So what I didn't... I'm doing the same thing here. What I didn't tell you yet is that we wanted to see whether this is specific or not. And it turns out it kind of makes sense that it's specific. This small RNA is involved in muscle metabolism, and what happens in ALS is there are lots of other problems, but one of those is muscle problems. So we have a whole bunch of cases of samples from people with Parkinson's and controls, and this thing is not elevated. So it's probably specific. We're now testing it to see if it shows up in Alzheimer's and some others, but we suspect that it's going to be specific for ALS. Now, I don't know how many of you have had experience with ALS. It's hard to diagnose every one of these diseases. I mean, it's often a diagnosis of exclusion. Eventually, they get it with the biomarkers that Nick will talk about for Alzheimer's much, much better. This is really helpful for ALS because sometimes you often go for a long time before you really know what's there. So we started the same thing in Parkinson's. Now we're doing that in our clinic over in this building across the street. This is led by Megan Cochran, who runs the clinic with our team of genetics counselors, as well as Anna Hurst, who spends some of her time with us, comes up from Birmingham. And it's exactly the same thing. Recruited people. We actually advertised for this. I think I wouldn't be surprised if some of you participated in this. I did, knowing that my father had Parkinson's, to see whether I was a control or not. We don't know yet, but the idea, and we actually had just wonderful response to this. Megan, we're trying to crank it back up again to get more participants, even though we have a fairly large number of them now. And if you can go online and see how to participate in that. So we've enrolled. This is actually a little bit. There are a few more than this now, but about 60 people with Parkinson's and without. And actually, unlike the ALS, we're actually testing protein biomarkers as well. The combination of looking for the small RNA and the proteins it's likely to be more powerful than doing just one. And we're actually have gotten DNA. Genomic DNA, so that we can actually sequence their genomes if we find something. So we're really excited about this. It's just starting in this. The week before Ben left, he prepared all of the RNAs and DNAs for us. So we're getting ready to do those final experiments. So then I'm about to end here. What did we do next? we're, a research institute. We do a lot of stuff, but for this to become a clinical and used in clinical practice needs to be a whole lot more. We believe that it's real because it's replicated both, and we're doing the same thing with Parkinson's. But for the ALS, what we think we knew, we don't know whether we'll be able to find this, but a big company like a pharmaceutical company, some of the big companies do diagnostic stuff as well, and they often now want to have the diagnostics to go along with their drug development for the reasons that I told you earlier. So we're starting to look for that. Would love to see that happen. Publication, you have to publish if you're going to do anything in research, and so we have a paper that has been submitted. We'll see how that goes. Anyway, I wanted to tell you about this. This is just, as I said, we do a bunch of other things. You'll hear some of these here, and come back next year because we'll have new stuff as well. So thank you.
Thanks so much, Rick. To put that into perspective for y'all, just a little recap. For a fast-moving disease like ALS, every single day matters. What Dr. Myers and his team are doing by identifying these blood-based biomarkers is essentially giving doctors a way to peer inside the body and catch the first early warning signs at a cellular level long before physical symptoms appear. That kind of early detection changes everything for how we treat diseases. If you have a question about how these ALS biomarkers are discovered or how close they are to being in the clinics, now is your first opportunity to submit a question. Like I described earlier, if you'll just pass your questions to the left, someone will come and pick those up. While those cards are making their way down the aisle, let's keep things moving with our next speaker. Our next speaker tonight is a graduate student in Dr. Nick Cochran's lab. She's doing her graduate studies at UAB, but her research project here at HudsonAlpha. It's my pleasure to welcome Ashlyn Anderson.
Hi, everyone. Today I'm really excited to share on some of the work that we've been doing in the Myers and Cochrane lab on studying microglia and neuroinflammation. Okay. Oh, there we go. Now I'm really loud. Yeah, so some of the work that we're doing to study microglia and neuroinflammation. So to start, I just want to give a little bit of information on what microglia are and why we care about them. So when you think about cells in the brain, you've likely heard of neurons, and Rick mentioned it a little bit, but the brain also consists of a lot of other cell types. And the neurons are really important because these are the ones that are signaling in the brain, but there are other cell types that are also really important. And the brain consists of other glial cell types, such as microglia. And microglia are our brain's resident immune cell. And so you can kind of think of them as our brain's defense and kind of cleanup crew. And these microglial cells are constantly surveying the brain to look for signs of infection or injury. And they also are capable of taking up material and clearing debris and dying cells from the brain. So these cells are really important for maintaining a healthy, clean, and functioning brain. Okay, and so these cells are important for the healthy brain, but they're also involved in a process called neuroinflammation. So when we think about inflammation, we usually think about it in the context of other parts of our body. So for example, like if you cut your finger, your immune system responds, and that immune response is really important for preventing infection and starting tissue repair. And a similar thing happens in the brain, where our microglia can detect when there's potentially injury or abnormal material, and they can go from a resting, surveying state to becoming activated, where they can then clear debris and dying cells from the brain. And this is really important in neurodegenerative disease because when we think about these different neurodegenerative diseases, they often have different protein aggregations that are characteristic of these different diseases. And specifically, for example, in Alzheimer's disease, you have plaques that aggregate in the brain. And this can impact normal brain function, but it also can signal to microglia that there's something wrong. And in the short term, this response can be really helpful because those microglia can help clear this material. However, in neurodegenerative diseases, this process can end up becoming more long-lasting, where instead of just having a short cleanup response, you end up with more chronic inflammation. And that chronic inflammation can end up being detrimental to the brain over time. Where over time, this inflammation ends up impacting nearby cells and can actually end up leading to further neuronal damage and neuronal loss and accelerating cognitive decline. So when we're studying these neurodegenerative diseases, it's important that we're not just looking at the neurons, but also looking at these microglia cells to understand how they're going from when they're in a neuroprotective state versus when they become overactive and potentially lead to more chronic inflammation and neurotoxicity. So one of the challenges is that these cells are also really hard to study. So microglia are in the brain, which means that we can't easily collect these from living patients. And we can use animal models to study the brain, but these don't fully capture human genetics or human biology of microglia. So one of the ways that we've been studying these is to use induced pluripotent stem cells, or iPSCs, and the basic idea with these is that we can take cells from someone, usually like a small skin sample, and then we can take those skin cells and reprogram them into a stem cell-like state. And once they're in this stem cell-like state, we can then guide them to become a bunch of different cell types. And if you've been to one of the M&M events in the past, you've likely seen some of our work where we've been able to take these iPSCs and make them into human neurons. But more recently, we've also been working on making these iPSCs into human microglia. And this is a really powerful tool because it gives us a model of actual human microglia. And then we can grow millions of these cells in the lab and test different perturbations and under different conditions to see how these microglia change. So for example, we can look at microglia in a resting state, which is similar to how they would be when they're like surveying their local environment, or we can treat them with a stimulus that drives them to that more activated microglia state. And this is going to be more similar to what we might see in neuroinflammation or potentially in disease. And so we can study between these two different conditions to look at how this activation might be relating to neurodegeneration. And so once we have this model, like the next question is, what can we do with it? And one of the ways that we've been using our model of microglia is to look at genetic risk factors for neurodegenerative diseases, specifically for Alzheimer's disease. And a lot of what we know about the genetic risk factors for Alzheimer's disease come from large population studies where they've looked at the DNA of individuals with Alzheimer's disease versus those without, and they've been able to identify regions of our DNA where specific letters are associated with an increased risk of disease. But these are just associations, and we still don't know a lot about what these regions are specifically doing and how they might be influencing disease risk. But we do know that a lot of these seem to be really important for regulating or controlling genes specifically in microglia. So we've been using our model to study what these disease-associated regions are actually doing and what genes they're controlling. And then we can also evaluate how they're impacting microglia function. And one of the ways that we can assess this function is here on the screen. So this is showing what cells have specifically taken up debris. So like I said, one of microglia's main functions is to take up dying cells or debris from their environment. And in red, we can see the cells that have already taken up other material, and this allows us to assess microglia function in a more functional way. And this is really exciting because it's allowing us to build on these other studies where they've already identified these disease-associated regions, but we're taking this into another step where we can further characterize what these regions are actually doing and how they might be impacting microglia function, and further kind of decipher how disease mechanisms may be happening. And this also allows us to kind of connect human genetics with how actual biological changes that are happening in our brain's immune cells. And lastly, just want to finish with saying that some of this work where we were characterizing this microglia model was published just this week in iScience, so really exciting for both of our labs. Thank you.
Thanks so much, Ashlyn. Like Ashlyn said, because we obviously can't peek inside a living person's brain to watch a disease develop, scientists have always faced a massive hurdle in studying these conditions. What this work is doing is completely changing the game. By using stem cells, Ashlyn and the team are essentially hitting the rewind button on standard cells and growing living human brain microglia right in the lab. How cool is that? It allows them to create a window into the brain, watching in real time how genetic changes overlap across different diseases so we can stop them at the source. You guys look like a curious audience. I know you have to have questions after this presentation, so if you have a question for Ashlyn or any of our other speakers, please pass them to the left right now. While those cards are moving down the aisle, I would like to introduce our final speaker of the night. If you've been at an M&M event in the past, you probably know him. It's my pleasure to introduce now HudsonAlpha faculty investigator Nick Cochran.
Thank you so much. It's good to be here with all of you tonight. So I am, my goal tonight is to give you an update on the HOPE-AD project that we've been doing. And I know you've seen a lot of slides tonight, a lot of the highly technical data of some of the techniques that we do in the lab. And Ashlyn did a really amazing job of talking about some of the new things that I know you haven't heard about because it's new technologies that we're implementing in the lab. But one thing that we always are trying to think about is how do we take what we do in the lab and then translate that to impact, right? And there's a lot of things that will eventually have impact on a population level, and that's something that we always are thinking about as scientists. But it's also great when we can do something that's really directly impactful, and even better when we can do that in a way that impacts our community right here in North Alabama. So that's what we've been trying to accomplish with this HOPE-AD project. And it's been going on for a little over a year. Just to remind everyone, the goals of this project are to really accelerate the early detection of Alzheimer's disease risk so that we can get that information to folks and have that information available so that they can do something about that risk level. For a long time, that hasn't been possible. There just weren't very effective treatments or things that you could do with lifestyle to move the needle. But we now have established interventions in both of those categories that we can recommend. Now, don't get me wrong, these things are not cures, and we have a long way to go. But we do know that there are some things that we can do, and there is hope. You've heard about that some already tonight. So in the past year, we've made a lot of progress on this project, and I really have to give all the credit to the clinic team. So Megan Cochran, who does happen to be married to me, and her and her team have done an amazing job in implementing this, and really, but all of you, and I say that knowing that there are potentially some participants in the room. Please don't raise your hand or jump up. But the community here is also critical because we were able to hit our enrollment goals really quickly for this project. So all in all, 165 people came through the doors of the Smith Family Clinic to participate in this study. And in order to participate, you had to have a first-degree relative who has or had some kind of dementia. And then you were assessed by our team for several other things, right? So you're evaluated on family history, on certain genetic changes, including ApoE, and other non-ApoE polygenic risk scores. That's a complicated term, but it just means kind of the rest of your genome and the contribution of that to Alzheimer's risk. And then also on key biomarkers. Rick mentioned some biomarkers in development, and that's really important work that we're doing here at the Institute. There are also good established biomarkers for Alzheimer's now that you can measure from a blood test. And that's something that we're capitalizing on in this study by measuring those biomarkers. We're also doing lifestyle and health surveys. We know that there are things, and it's not a secret, it's the things that your cardiologist and general practitioners have been telling you, but all of the things about mind diet and about exercise, all of those things really do make a difference when you consider them all together on disease risk. By looking at all those factors, we were able to identify 52 individuals who are at particularly high risk. And for those individuals, we invited them to participate in a multimodal lifestyle intervention. So to, with dietitian guidance, shift as many factors as they could to the healthy category. Now, in our population in Huntsville that's highly educated, a lot of people were already doing a lot that was really good, and that's a great thing. But we did move the needle on some of those other factors. And that actually concluded just today. So the last two participants came to Smith today to finish that 6-month visit, but we're not done. So we've gotten some great data from that, but with philanthropic support, we were able to add some things that we weren't sure we were going to be able to do when we started this. We were able to get a retinal scanner for the clinic. There's good biomarkers in development looking at retinal scans. This is something that could be implemented in the developing world where some of these complicated tests may not be available. We were able to extend to 12 months. So now we're inviting people back to gather even more data on how things change over time. And we're also able to start piloting something called a Tasso device. This is a way to collect a really small amount of blood in a very non-invasive way by putting a device on your arm. It's a very small amount of blood. And the other thing that's really important about that, and hold for future updates, this is something that we could put in an at-home testing kit. So we're really excited about that as a future direction. So just to say a little bit about the results, and we're still, we still got to dive deep and analyze the data because, like I said, we just got the last 2 data points today. But with a preliminary look, what we're seeing is that, over the 6-month period, and we'll get a 12-month, and we've got to keep looking at this over time. But even over a 6-month period, we saw that these healthy habits, they stuck for folks. We were able, with dietitian guidance, to really promote those healthy habits, and we have the data to back that up, where we have fitness tracking and that sort of thing. Their metabolic health also stayed steady, and importantly, we are seeing signs that the folks who adhered better or improved those factors had less decline on their cognition scores, right? So that's something that's really exciting to see, that we're starting to see some signals in that of things that other folks have seen in the field with similar studies, and it shows that you really can do something with multimodal lifestyle interventions. Again, not a cure, but something that does move the needle. The other thing, and this is really important, with the small numbers that we have, we knew that we were really positioning ourselves to do more in the future. And so please do hold, because we're going to have a lot more to talk about in the coming years. But one thing that we could assess with the numbers that we had is attitudes towards receiving this kind of information. This is something that's actually still very controversial in the field, is, this is high-powered information. Do you really want to know this information? And the surveys that are theoretical in nature, that say, like, if you had this information, would you want to know, tend to say that people do. And of course, if you don't want to know, you just don't have to do it. But no one's really looked at all of these things in combination and then said, okay, how are you responding to this information after you actually receive it, right? And what's really compelling about the data that we have so far is 85 to 90% of people, they do just fine with this information. They've probably already built kind of an idea in their mind on how they would respond to this, right? And so we're really encouraged by that. And the people who didn't, it turns out that most of them had some pre-existing either anxiety or depression that's likely contributing to that response. So that's something that needs further study, but we're excited about the fact that we think that we can roll this out in a safe way, that people, are going to be able to absorb this information, even though it is, of course, very impactful information. So with that, I will stop and get on to the next section. Thank you so much for being here and for your attention.
Thank you, Nick. What I love about the Hope AD project is that it represents a massive shift in how we think about brain health. For a long time, medicine has acted like a fire extinguisher, waiting for a fire to start before trying to put it out. But what Dr. Cochran's work is showing us is that we can act like a smoke detector instead. By looking at the intersection of our genetics and our daily habits, their team is building a blueprint for how we might prevent Alzheimer's disease from ever taking root in the first place in the future. If you have any questions about anything you heard tonight, this is your last chance. Please pass them to the left. And while y'all are doing that, we're going to get set up for the Q&A. So I'm going to invite our speakers back up to the stage, and we will be right back. Okay, we're going to get started with our first question. First question is, I feel like I have seen more and more news about wins in neurodegenerative diseases over the past few years. Why does it seem like progress in neurodegenerative disease research has been ramping up lately?
You want me to start? Okay, so that's a great question, and I feel that way as well. And I think that there's really just been a constellation of breakthroughs over the past several years that have really increased enthusiasm. And so that involves kind of three of the things that you've heard about already tonight. And it's some of the toolkit that we're using and now trying to implement and accelerate, right? So the first is new treatments. We have been trying as a field to get new treatments for a really long time. And we are finally there. We have new amyloid immunotherapies. So this goes after one of the key pathologies. It's not a cure, but when administered in the early stages, it can slow progression by about 30%. That's tangible. That's real life years. And certainly real years that you can delay some of the most severe symptoms from occurring. So this is really exciting, and there's more to come in that area as well. I think it's accelerated investment in some of the secondary targets, things like tau, that are now in clinical trials. The second category is these early detection biomarkers. And so that's, again, something that we're implementing. It gets even more complicated. It's not just, and this is now specifically for Alzheimer's disease, it's not just that one that we're using in this study, but actually different flavors of that can tell you about staging of disease. And this is work that's just really been happening over the past 6 months to a year. So now there's good biomarkers for pre-symptomatic / right at the edge of disease, for when someone is kind of in the middle of mild cognitive impairment, and then for when someone has reached the stages of frank dementia. And those all can be very characterized by progression of these three different biomarkers that are somewhat related. So we're thinking about how we implement that as well. But again, that's just over the past year. The final piece is these lifestyle data, right? So there's a study in Europe that's now been going on for almost 10 years, where there was promising data about lifestyle, but of course we had to do it in the U.S., right? And so we did, and there was positive data from the U.S. as well. So now it's really exciting data. There's also similar efforts in Australia, and it's spun up ones in South America and all over the world, saying that multimodal lifestyle interventions really can make a difference. You know where they've not done it yet? In the Deep South. And so that's a place where we can make a big impact, right? We know that when you're talking about multimodal lifestyle interventions, diet and exercise, we've got a lot of room for improvement. And sometimes that gets giggles, but it's true, right? And so I'm really excited about being able to accelerate that as well. So I think that all of those things together, it's really built enthusiasm in the field.
Let me add to that. You can hear me, yeah. So this is a general statement. First of all, that's insightful. It is really changing. The last few years have been extremely exciting. This is the way science works for almost everything. You get accumulated knowledge, and then it builds up and everything is built on lots and lots of other people doing stuff, no matter what you say, what happens. The other thing is that this also, I'm old, I've been doing this a long time. You see this in science where something is puttering along and somebody invents a technology or a bunch of technologies, and then it goes up, and that's happened a lot over my career. I'm not responsible for those, but I've taken advantage of them. One of those is genomics, the fact that we can sequence a genome in an afternoon for a few $100s as opposed to to several $1 billion over 13 years, which is what the first one did. And that's true for imaging and many other things as well.
Thank you. I think we can all agree that we have seen the term AI everywhere. So the next question is, how is AI helping you all do your work faster, more efficiently, do better analysis, etc.? Ashlyn, I know you're a computational gal. You want to take that one?
Yeah, I guess I'll start with that. So I am mostly a computational biologist, which means that I do most of my work at a computer, and we deal with a lot of really large data when we're studying these diseases. So we need really high computational approaches for analyzing this data so we can kind of pull out and decipher the information that we're getting from it. And we're all, and me as well, we're using some of these AI tools to kind of speed up this coding process and help us debug some of our scripts. So we have experience with coding. I learned how to code before AI was in existence, but this is helping us by actually speeding up how much time we're spending writing these scripts and helping us catch things in how we're analyzing the data.
Yeah, so I'm going to add to that a little bit. So, this is everywhere, right? And it's in every industry. A year ago, I may not have had much of an answer for how we're using it at HudsonAlpha, but now I do. And before adding to Ashlyn's answer, I want to say something about HudsonAlpha particularly. We are really good at being nimble and adopting new technologies quickly. And that is something that I'm seeing. I talk to folks in the field who are in a variety of places, and they're asking what we're doing with this, and are really impressed with how quickly we've been able to reach an institution level adoption of some of these tools, and they're running into clunky things. I won't say the places that are struggling with this, but anyway, we're really good at that. So know that it's not just sequencing technologies and other technologies that we're good at adopting. We're really adopting these AI tools as well. So beyond the answer being that I go to one of my really talented folks and say, how can we use AI? We are using it in a couple of other ways that I'd like to point out. So on the computational side, Ashlyn mentioned that, and I'll give one example, or two examples actually, of how we've done that. And one is also an example of how it's really able to accelerate career growth and development for folks. So we have someone who was just in our lab, just finished up, and had a plan to come to the lab for about a year and then go to graduate school, and that's exactly what happened. He had grad school offers from a dozen places, and really excited that he's accepted and about to start. But one thing that he wanted to do, he said, I want to learn some computational approaches, and I'd love to be able to implement some of these new AI tools. And that's exactly what he did. So we have a preprint up now. It's something that we've done just over the past year, and he did it. He learned how to use it. He used a tool specifically developed for understanding the effects of genetic variation on a gene that we care about, and implemented it, and now that is out there for the world to see. and was able to do that just in a period of a couple of months. So it really accelerates the progress of what you're able to do. Another thing that we're able to do, so some of you may have heard Greg Cooper talk about childhood disease and how important reanalysis of genomes is. And so this is something that we're thinking about AI for as well, and it's a place where we've seen some advances in neurodegeneration. There are times where we see some people and we're like, you know what, there's got to be something genetic going on. And Greg Cooper talks about this with childhood disease a lot. Sometimes it happens in neurodegeneration as well. We had a couple of siblings from South America, and it's like, this has got to be genetic. And they actually had one genetic variant in a disease where it's recessive, where you need 2 different changes. But they could not find the second variant. They just couldn't find it, so we kind of put it in a drawer. We said, well, we'll come back to it, and we came back to it with some of these new AI tools. And the first thing that popped out, the most significant thing, we looked at it, we looked at what it might be, and it was like, this is it, this is the answer. So again, that's something that we're about to put out as a preprint and then to publication, and we needed the tools to be able to do it, because there was just simply too much information there to sift through, but we were able to sift through it with some of these new tools. So those are some of the ways that we're using AI. It's really making an impact and getting us to new insights that we can share with the world.
Add one quick thing that came here from Stanford. My wife and I did 18 years ago to help start HudsonAlpha. Great place for computing, et cetera, but there was something really appealing about this town, which is, I don't know, tens of thousands of engineers and computer scientists. And I love bragging to them back there that we have real rocket scientists working in our labs. So. And we do.
Nick, you mentioned fundraising for the retinal scanner. There is a question about how is the retinal scanner being used in the HOPE-AD study?
Yeah, absolutely. So the retinal scanner, so what we're using that for, there's research going on how retinal scans can correlate with some of the key pathologies in Alzheimer's disease. And so it's currently what we're using it for is we're doing these retinal scans every time the high-risk participants come into the clinic. So it can help us track both abnormalities that may be related to the pathology, but then also how that progresses over time. What it could end up being is something that's even less invasive than these blood-based biomarkers as a way to track and a way to catch things in the population, right? So you could imagine opting in and checking a box when you go to the eye doctor, for a screening test for Alzheimer's disease. And that's even less invasive than having to go get a separate blood test or a finger prick or anything like that. It's just like checking a box that, sure, I'd be interested to know about that, and then you can go get a more detailed test if that screen comes up. So it's just one more way that we can accelerate early detection of these diseases.
Thank you. We've talked about a little bit throughout each of your talks, but the question is, what are the meaningful treatments, if any, today for dementia and Alzheimer's disease, and are there any in the pipeline? Sure.
All right. So yes, so meaningful treatments, and this is something, there's been a couple for a long time that still are meaningful in the form of Aricept and Namenda, and they will plateau symptoms for about 6 months each. They'll, in combination, delay nursing home placement by about 2 years, and that's meaningful. It is not a cure by any means, and they are not disease modifying. They are addressing symptoms. There are also, now there's a new approved drug for agitation for Alzheimer's disease, and then in the disease modifying front, that's the real advance that's happened in the past few years. So now there's 2 that have similar effects that are these immunotherapies. They either are administered through an infusion, like at an infusion clinic. Now there are some that can be administered at home with an injection just into the stomach. So that's really helped to accelerate implementation of these. So they're rolling out their disease modifying. Now those are most effective in the very early disease stages. So it's just another reason why early detection is so important. And again, even in combination, all of those things plus a multimodal lifestyle intervention, we're not to a cure. So, these diseases are going to keep progressing, but we do have a way to slow them down now.
Maybe this is more hope but I think it's based on reality too. That acceleration in knowledge has made a difference, a huge difference. For any disease, if you know which targets to go after, and that usually means a protein or a set of proteins, if you know which targets to go after, that's what most drugs go in and they bind to those targets and stop them from doing the bad stuff that they do. And that comes from really basic research, understanding the biology and the underpinnings of everything that we do. That's what's accelerating too, and hopefully it won't slow down. It requires real long-term support. And I'm not talking about here, I'm talking about nationwide and worldwide to gain that kind of knowledge. And already we're seeing this. I think Nick taught me a lot about Alzheimer's. I worked more on some of the others, but most of the treatments that were drugs that were being developed, that many that fail, were going after the same target. That is an important target, but now there are other things as well. And I think about that with regard to the other diseases that we're working on as well. So I really do think we're going to see more, and it would be great to not just give extra years of life, but to end it altogether.
A related follow-up on that, we do mostly basic research here at HudsonAlpha, but we definitely have major impact. How is, for example, the work from HudsonAlpha reaching companies and other research labs? The question is there a national forum where this work is being shared, or how does collaboration like that happen?
One of the things that Jim Hudson and Lonnie McMillan wanted to do that was kind of crazy. Let's have companies and basic academic style research in the same, not just the same campus, same building. And initially, on the main building, the companies were going to be on that side and the academic labs are going to be this side, and we've actually intertwined them. We have to pay for them differently. We're the landlord for those companies, but we bump into each other, eat lunch together, do all sorts of things. So that has been a tremendous plus here. We started with 5 companies. We have 52 now, and they're growing. We have a fellow named Dylan Fritz who joined not quite a year ago, who is running that program, and it is on steroids now. It's really exciting to see. That's just what we're doing here, but you do that throughout the. I mentioned about the ALS biomarker. We're not going to develop that into a clinical biomarker here. It requires, can't even imagine how much money it's going to require. It's going to require thousands and thousands of people. A big company that does that is going to need to do it, but we then transfer that kind of technology. This young lady here did that kind of tech transfer in Atlanta right before she came here, so she helps to participate. Sarah helps to participate in that as well. And the main reason for doing that is to speed it up, and that's why Jim and Lonnie wanted to have them in the same building, so same campus.
Thanks, Rick. I have 2 more questions. The first is related to participation in research studies. So can one of you speak on how people can participate in future studies, both here on our campus and just generally in clinical studies about dementia and Alzheimer's?
Yes, I'll take that. So for the ones on our campus, so first, we are not currently enrolling, for example, for the HopeAD project. There is still open enrollment for the Parkinson's project, but that, you just got to look at the enrollment requirements for the study. They do take both patients with Parkinson's and healthy controls, but there are specific requirements, and they may be enrolling for one or the other at any given time. For studies like HopeAD, when we do have an opening of the enrollment, and we do plan to do more in the future, there will be public advertisement of that with the criteria of what is required to participate in those studies. That's going to be similar through other venues as well. So there are things like the Alzheimer's Association has good resources, a resource called Trial Match, where you can put in what characteristics are available and then narrow down on studies that you may be able to participate in. There are also studies like we work collaboratively with UAB's Alzheimer's Disease Research Center that tracks people longitudinally. So that's another possibility, but they have specific enrollment requirements. So it is study specific, but there are some of those resources like Trial Match that can help you find studies that you may qualify for if you'd like to contribute, even including as a control.
Thanks, Nick. For the last question, I would like each of you to answer, if that's okay. What are you most excited for going forward with either the research we're doing here at Hudson Alpha or just progress in the field in general? You go.
I should have prepared for it. I'm excited about a lot of stuff. I'm kind of blown away about how much faster things are moving. I think I said it on this stage maybe a year ago or maybe 2 years ago that 40 years ago I'd say, yes, we really hope we're going to be able to make progress on this, and we would, and it would be pretty incremental. And I wouldn't say I didn't have hope, but it always seems so distant in the future. We can see it now. And that's what, to me, is the most exciting part. I love the knowledge, the gain in knowledge. We're learning so many different things. Some of this, you might be studying this, well, for one thing, when you study this brain disorder, it impacts lots of the other ones as well, because some of the knowledge is and the pathways and things like that are similar. That's what gets me up in the morning, but it's also seeing where it's going and how rapidly it's going. We're not there yet, as Nick keeps saying, we're not curing. I'd love to see it be where you really end it, so we all would.
Okay. Yeah, so I guess we talked about it a little bit, but I think for me, one of the things that I'm most excited about is actually, like how we're implementing some of these AI tools, and we're getting more out of the data that we can look at, and kind of using these to better understand disease and accelerate what we're learning from our data so that we can put it on potentially on a faster timeline so that the discoveries that we're learning in the lab can get faster translated to the public and kind of build those discoveries faster using some of these AI tools. Yeah.
All right, so we've talked about a lot of things that are really exciting tonight, but one thing that is always exciting to me as a scientist, stepping back more generally, and certainly in the context of this room and the impact that it has on Alabama, is not just the impact on the participants that we've talked about, or all the exciting discoveries that we've talked about that are going to accelerate early detection, but also it's really exciting as a scientist to be a part of these projects that are much bigger than just you, right? So I'm most excited about what Ashlyn's going to do, and what Jared Taylor, who's a computational biologist in the room, is going to do, and what Bree Rogers, who's a senior scientist in my group, is going to do, and what Megan Cochran and her team at the clinic are going to do. It's really great to see folks who train in your group, what they do and go on to do, and that's workforce development. For here in Huntsville as well, right? We are employing really talented people here. We're in the process of hiring for a couple of roles, and we're getting hundreds of applications for each of these roles. So we have our pick of the most talented people in Huntsville and around the country to work on these problems. That's a really exciting place to be, and to find, people who are really talented, who are going to think about things that you never thought about, like Ashlyn does in my lab, about these problems. So that's what I'm most excited about for the future.
Let's have another round of applause for our speakers tonight. As I mentioned at the top of the program, this is my 4th M&M to moderate, so I've been staring at you all for 4 years, and I see a lot of new faces this year. If you are interested in learning more information about us or getting notified when we have more research studies, if you would just want to write your email down on a note card or get in touch with one of the advancement staff members, they can make sure you get on our mailing list to learn about all of the exciting stuff going on here. At this time, I would like to invite Russell back to the stage to close us out.
Thank you, Sarah, and thank you, Rick and Ashlyn and Nick. Great job to all of you. As you've seen, Sarah's a gifted communicator, and I stole a statement that she made, and she doesn't even know I'm about to say this. But she wrote something that said, "Discovery doesn't wait, and neither does the need for it. Philanthropy helps make sure the next breakthrough arrives on time." And so I think there's so much truth in that. And I want to thank each of you. So many of you have contributed to our Memory and Mobility program, whether that's one time or multiple times. We thank you because your contributions, your philanthropy, helps us make advancements in our research. Your generosity helps us to move forward, and what you see that our team is excited about, your philanthropy helps drive that and support that. So thank you for all of that. If you would be willing to make a gift tonight or in the coming days, there's a QR code on the screen that you can use. And as always, you're welcome to make a check to HudsonAlpha Foundation. But again, we want to thank you for that. And we also want you to know that your gifts, they matter to us because you do help us to advance research. And as part of that, we want to also share with you that we're so appreciative and for the gifts that you entrust us with, we will be good stewards of those gifts. I think you see that for what our team does, and I think you see the results that our team continues to drive. So thank you for any part that you play in that. We're grateful for your support, and again, we're committed to putting it to the best use. As we close, our team is going to be here at the front, so if you would like to continue any conversations or if you have questions, please come join them. And I'm going to look at my notes to make sure I get this right. Our next event is the Southern Solutions. It's Plant Science as an Economic Engine. And so that will be on August 18th at 5:30 on a Tuesday at Mars Music Hall at the Von Braun Civic Center. We hope you can all join us for that, and you'll receive details in the month of July for that event. But with that, I sincerely thank you for being here. Just looking out at this crowd, it's fantastic. Your passion for what we share tonight, your passion for Hudson Alpha, thank you. I want to wish you all a great evening. Thank you for your support. Thank you for the friendship that we have here at Hudson Alpha, and I look forward to seeing you again soon. Thank you so much.